Good morning. Myself Dr.mumtaz, Associate Professor, Department of Botany, Dr. Rafiq Zakaria, College for Women. Today I am giving the demonstration of the micro teaching. Structure and function of gene. Fine structure of gene that is given by the Seymour-Benzer. One gene, one enzyme hypothesis. Genes and related disease, that is phenylketonuria and alkapetonuria, detection of genetic disease, amniocentesis, genetic counseling. So first of all we shall see the fine structure of gene that is given by the Seymour-Benzer. Gene is a hereditary unit which is transmitted from one generation to another generation, and gene is a fundamental biological unit which plays a very important role. Mendel has already explained the result of his experiment in the monohybrid and dihybrid cross. First time the word hereditary factors or hereditary elements given by the Johansson in the 1909 used the term gene for the Mendel factor. The presence of the gene is detected only when a mutation occurs. Initially, the genes were considered as a bead and chromosome as a chain of bead. That has been explained by the scientist Morgan in the year 1911. Each bead was thought to control one character by controlling some biochemical step. There is a fine unit mutation. Development in the molecular genetics used to the fine analysis of the structure and function of the gene has given a new concept in the chemical genes. And the single base to stretch the DNA molecule, that recombinant can occur between two bases. This gene neither a unit of mutation nor of recombination. Hence, one gene, one character hypothesis given by the D varies, which was become absolute or incomplete sort. In the year 1923, Morgan, who was the first time demonstrated that the locus of the bar body in Drosophila contain more than one functional and recombinational unit. In the 1950, Rupert found that crosses between different biotin requiring mutants of Aspergillus can yield wild type recombinant. In the year 1950, Benzer reported fine structure of genetic region which was explained in the T4 bacteriophage, recognized three types of genes: cistron, recon, muton, which was followed by the E.B. Lewis in the 1952 and demonstrated that the white eye color locus of Drosophila can be divided into many functional subunits. This finding led Montecorvo 1952 to suggest that each gene has linearly arranged mutable sites between which recombination can occur. Cistron can be explained that is the two genetical units exhibit syntrans phenomena. They are considered to belong to the same functional unit which is called the cistron within the same gene and exhibit the different functional cistron. With the help of the recombination that has been given intragenic level, there has been gene further divided into small units called mutational sites. The white eye locus of Drosophila has been resolved in several sites. Likewise, the micromap of the Logen's locus in Drosophila has been given, that is genetic unit that has a single function. This has been defined as cistron in the equivalent of the functional gene. Next is the muton. There are many position or sites or angle within a cistron where mutation can occur. Therefore, the gene as a unit of mutation is smaller. It consists of very few nucleotides than a cistron. According to the Benzor, a muton is the smallest pigment of DNA which is capable of mutational change. Thus, a mutation can be defined as the smallest unit of genetic material which change or mutate produce a phenotypic effect. Mutation may thus be delimited to a single nucleotide or some part of nucleotide. Different forms of mutationally defined genes are called as a homoalleles. Example, in the bacteria, muton may be present, or even cistron for the hemoglobin. The muton can be single nucleotide. Next is the recon. The structure of gene as a recon is also discovered by Benzer, 1962. According to him, recon is just a subdivision like the muton. Again, the recon is the smallest unit of cistron or DNA which is capable of crossing over and recombination. Recon may be as small as one nucleotide pair in DNA and one nucleotide in RNA. Recon and muton are usually identical, and it is a small piece of DNA polynucleotide strand which codes for an polypeptide. Millions of genes are present in a genome of an individual cell, but only 30,000 actually codes for various structural and enzymatic proteins. Now we shall see about the one gene, one enzyme hypothesis. This has been proposed by the Biddle and Tatum in the year 1948. He has explained the synthesis of one enzyme, that is protein, and this concept is called as one gene, one enzyme hypothesis. After the discovery of this, he was awarded Nobel Prize in the year 1968. They did their experiment in the neurospora and red mold, which is a member of Ascomycotina. The neurospora crassa can grow in minimal medium containing sugar, salt and biotin in the laboratory or also in the naturally. It can synthesize all substance like protein and nucleic acid from the medium. Neurospora synthesizes arginine from glucose. This metabolic reaction involves many steps, and that step is catalyzed by a specific enzyme. That is, the Biddle and Tatum treated the spores with X-rays which can cause gene mutation. X-rays treated spores in variable minimal medium, and some of them grow in the arsenic content, and third group of the spores grew in the medium containing ornithine. This experiment shows that the first group of spores are unable to synthesize arginine because they are able to produce the enzyme to convert the citrulline into arginine. This is because the X-ray caused mutation on the gene producing EN1. Similarly, the second group of spore could not synthesize citrulline because they are unable to produce the enzyme EN2 to convert the ornithine into citrulline. The third group of spores could not synthesize ornithine because they are unable to produce the enzyme EN3 to convert the glutamic acid into ornithine. This experiment proves that enzyme is controlled by gene. When the gene is changed, corresponding enzyme not produced, so there is a block in the metabolic reaction. So this has been given as a one gene, one enzyme hypothesis. In the year 1908, Garrod described the inborn errors of metabolism. After knowing the metabolism, there are some of the errors. They include phenylketonuria, alkaptonuria and albinism. These metabolic defects are explained on the basis of one gene, one enzyme hypothesis. These diseases are example of biochemical blockage. The dietary protein are hydrolyzed in body step by step and finally completely hydrolysis and converted into CO2 and water. Proteins are first converted into phenylalanine. Tyrosine is another amino acid, then phenylalanine is converted into phenyl hydroxyl phenyl pyruvate and then converted into homogenistic acid. Then it is finally converted into aceto acid. Then it is converted into water, CO2. This process is called as a phenylalanine metabolism. That is, conversion has been given here, how the child caused the disease called PQ idiots, because in the person phenylalanine hydrolase absent, there is a DNA mutation occurs. This is a biochemical pathway blockage through production of enzyme, and when the gene is absent, there is abnormality occur. The human phenotypic trait characters are affected by hereditary. You can see that physical appearance such as height, facial feature, eye color, skin color and hair color. Heredity also determines biochemical conditions such as genetic disease. Inborn errors of metabolism, gene produce enzyme whereas a mutant form these do not produce them, hence resulting in the physiological abnormalities. In metabolism of phenylalanine, it is an essential amino acid, and such as there is a showing about the breakdown of the phenylalanine, and 3 diseases are associated and they are given as a phenylketonuria, alkaptonuria and albinism. So you can see here the phenylketonuria, which is also called as a autosomal homozygous recessive. PP fails to produce the phenylketonuria, which is damage the brain tissue, and there is a phenyl pyruvate in the cerebrospinal fluid. The abnormal gene is suppressed here, and this is giving the disease known as phenylpyruvic idiot, that characterized by mental defect, feeble-minded, hypertonatry, light pigmentation, and they are showing accumulation in the blood and cerebrospinal fluid. Next is the alpha-ketonuria. It is also the inborn error metabolism, hereditary disease characterized by the recessive gene. As a result here, homogenistic acid accumulates in the blood. Urine of such person turns black when exposed to air. The excess acid is attached to the collagen of cartilage and connective tissue, due to which the ear and sclera are stained black. Next is the albinism. This is a hereditary defect where the melanin pigment are absent from the skin. Hence, affected person are pale in color and their hair and eyes are brown in color. Such person called albinos. The albinos are highly sensitive to the light. It is the inborn error in metabolism. It is a hereditary disease caused by a recessive gene are present in the enzyme tyronase can be produced. Hence, tyrosine cannot convert it into melanin pigment.